Imagine the construction of a grand city where each new building mirrors its blueprint exactly, while the select few exhibit unique, innovative designs. This architectural dance of replication and variation mirrors the cellular wonders of mitosis and meiosis. Mitosis, the master builder of our bodies, creates precise replicas, ensuring every cell in our being is a perfect copy of its predecessor. In contrast, meiosis, the artist, crafts diversity in the blueprint of life, producing the extraordinary variety seen in the next generation. Together, these processes orchestrate the symphony of growth, repair, and reproduction, fueling the essence of life itself. Mitosis is a highly regulated and precise cellular process critical for organismal growth, tissue repair, and asexual reproduction. During mitosis, a single somatic cell undergoes a series of meticulously orchestrated steps to ensure that each resulting daughter cell inherits an exact copy of the parent cell's genetic material. This complex sequence involves the duplication and segregation of chromosomes, organized into distinct phases: prophase, metaphase, anaphase, and telophase, followed by cytokinesis. Mitosis guarantees that each daughter cell contains the full complement of chromosomes, maintaining genetic continuity and stability across cell generations. Join us as we delve into the stages of mitosis, where cellular machinery executes the remarkable task of faithful genetic replication. Before the actual process of mitosis begins, the cell undergoes a preparatory interphase. This is the preparation phase where the cell grows and replicates its DNA. It consists of 3 subphases: G1 phase, GAP1, cell growth and production of proteins and organelles; S phase, synthesis, replication of DNA; G2 phase, GAP2, further growth and preparation for division. The interphase is the preparation phase for mitosis, and it is also the longest phase in the cell cycle. The interphase takes place in the cytoplasm and the cell nucleus. During the interphase, the nucleolus and the nuclear envelope are distinct, and the chromosomes are in the form of thread-like chromatin. During the first stage, the prophase, chromatin condenses into visible chromosomes. Each chromosome has 2 sister chromatids joined at the centromere. The mitotic spindle begins to form, and the nuclear envelope starts to disintegrate. During the second phase, the metaphase, chromosomes align at the cell's equatorial plane, known as the metaphase plate. The spindle fibers attach to the centromeres of the chromosomes. During the third phase, the anaphase, sister chromatids are pulled apart by the spindle fibers and move toward opposite poles of the cell. Each pole ends up with a complete set of chromosomes. During the fourth phase, the telophase, chromosomes begin to decondense back into chromatin. The nuclear envelope reforms around each set of chromosomes, resulting in 2 separate nuclei within the cell. Cytokinesis is the physical process after the end of 4th stage of mitosis. The process is that of cell division, which divides the cytoplasm of a parental cell into 2 daughter cells. The cytoplasm divides, creating 2 separate daughter cells. In animal cells, this involves a cleavage furrow that pinches the cell into 2. In plant cells, a cell plate forms to divide the cell. Mitosis is a fundamental process that ensures the continuity of life at the cellular level. Through a precise and highly regulated sequence of events, prophase, metaphase, anaphase, and telophase, followed by cytokinesis, mitosis accomplishes the exact duplication of a cell's genetic material into 2 genetically identical daughter cells. This process is essential for growth, development, tissue repair, and asexual reproduction. By faithfully replicating and distributing chromosomes, mitosis maintains genetic stability, allowing organisms to thrive and adapt. It stands as a cornerstone of cellular function, underscoring the intricate balance of replication and renewal that sustains life.